Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show today, bringing you another really interesting guest involved in creating a better tomorrow and do some really fascinating research. Today, we have the honor of being joined by Dr. Thomas Metz, who is a senior scientist, a laboratory fellow, as well as principal investigator in the integrative Omics group within the Biologic Sciences Division of PNNL, the Pacific Northwest National Laboratory, where his research focuses on the development and applications of high-throughput metabolomics, high-throughput lipidomic methods in conjunction with proteomics to answer a variety of unanswered biological questions. Currently, he is the director of the Pacific Northwest Advanced Compound Identification Core within NIH Common Fund Metabolomics Program. He is a co-PI of the Proteomics Laboratory for the Environmental Determinants of Diabetes and the Young Consortium, lead of PNNL's new M over Q, or Mass Over Charge initiative, and an adjunct faculty member in the Department of Biochemistry and Biophysics at Oregon State University. He has a background, a Bachelor's of Science in Biology, Frostburg State University, a Bachelor of Science in Chemistry, San Jose State, and did his Ph.D. in Chemistry at University of South Carolina, and his work has resulted in a couple hundred publications to date. And just as a little review, the Pacific Northwest National Laboratories is one of our United States Department of Energy's national laboratories, managed by the Department of Energy's Office of Science, and they're involved in advancing a wide range of frontiers of knowledge, not just energy, but a variety of other areas, including chemistry, earth sciences, biology, data sciences, all central to their scientific discovery mission. A lot of really interesting themes to get into today. We're honored to have him with us. Dr. Thomas Metz, thank you so much for taking the time to come on the show today.
Thank you, sir. Thank you for having me. Glad to be here.
It's great having you. So I would love to start off at the beginning, because I was really intrigued. I took a look at your PhD, which was on this advanced glycation end product work, working with pyridoxamine and lipid peroxidation. And I found this so interesting, not just because of sort of the application, or the ability to interfere with AGEs and a variety of diseases, but the fact that sort of the advanced glycation end product space sort of brings in a lot of what we're going to be talking about. It connects the glycome, the proteome, the lipidome, secondary metabolites, and all sorts of other neat things. Talk about a little bit about the early days. Talk about your PhD here. I think this is an interesting way to get started.
When I started graduate studies at University of South Carolina, you perform the lab rotations, and you spend a little time in each PI's laboratory to learn the type of science they do, the type of person they are, the type of mentor they might be for you. And I, going into graduate school, had always had a keen interest in disease research. And so I made a few rotations through the PIs, and then I entered the laboratory of Dr. John Baines at the time, and he studied the role of Maillard chemistry and AGEs and the development of diabetic complications in aging. And I found it completely fascinating because it was, for me at the time, the interface between chemistry and biology. And the others at the time that were performing disease research in that department, were more focused on the biological aspects of disease, less so on chemistry. And so I found what Dr. Baines was doing to be really unique at the time in that department. And I remember sitting in his office one day, and he said, you know, I've got good news and bad news. The good news is I don't expect you to work any harder than I do, but the bad news is I work my tail off. He used a different word, but immediately I knew that that was the place for me, because that was sort of the approach that I liked was, tenacious and aggressive to, trying to understand the science of the laboratory. And so I joined that lab, and my role was to evaluate this compound that you mentioned, pyridoxamine, and its potential to inhibit the reactions between glucose, which is blood sugar, and products of lipid oxidation, which are reactive and will bind to tissue proteins and eventually make their way into a non-reversible form that will then accumulate. And so the hypothesis of the day was that these types of AGEs will lead to chronic, cumulative, and irreversible modifications of tissue proteins and affect their function and turnover and lead to diabetic complications. And for sure, there was a correlation in animal studies and even in humans. As one has diabetes and has uncontrolled or poorly controlled blood glucose levels, even high levels of lipids, these AGEs will be higher in such individuals than in those that might still have diabetes but just have better control of their blood sugar and lipids, and in turn those individuals are even higher than healthy individuals. And as the other fascinating aspect about this chemistry is that it's the same chemistry that occurs when we cook food.
Yep.
And so these compounds are in food are responsible for the flavor and texture and aroma of food, and yet we as humans have the exact same compounds forming in our bodies, just naturally. It's a non-enzymatic process, but it still forms naturally just based on chemical principles. And as John Baines liked to say, humans are ovens. We just have lower cooking temperatures and a longer cooking cycle compared to what we do with food. But yeah, fascinating chemistry.
Absolutely. And again, talking about that sort of that overlap where we may find as you focus on these proteins and lipids and secondary metabolites and so forth. Obviously, you've been heavily focused on human health, but looking now, taking a step back at sort of PNNL's mission,
Mhm.
and as mentioned in the bio, it's a lot more than energy. I mean, you have sort of broad swaths
Yeah.
of intellectual focus there, and as you were just saying, these lipids and proteins, and they're found in us. They can be found in those plants over there, that microbiome sample there, and so forth. Why is PNNL interested broadly in the omics space? Talk us, reintroduce us to PNNL
Sure. Yeah.
and a little bit of its mission there.
Yeah. I mean, I think the interest in omics goes back probably 30 years or more at PNNL, and it really originated in the pioneering work of Dick Smith at the laboratory in developing mass spectrometry-based capabilities to measure biomolecules. He was at the forefront in the early days in all aspects of such measurements, focusing on ionization, which is the first step in introducing a molecule into the instrument, and then focusing on the separations of the molecules inside the instrument, their detection, and really he was one of the right people in the right place at the right time. Very early days in such types of research and measurements, and he really led a strong team at that time and capitalized on the knowledge and the technology being developed at PNNL in his group to show the utility of such measurements in proteins, metabolites, or lipids. And we just institutionally built off from that through the years. I joined the group 20 years ago as a postdoc, and I stayed as a staff scientist after that. And we pushed the funding agencies to consider these technological developments and routine applications relevant to their mission areas. And we had a lot of success, of course, in proposing these techniques to the NIH, but also to Department of Energy in understanding bacteria, either bacteria that were in isolation. There was an early project that was looking at a bacterium called Shionella onidensis that was capable of taking uranium that is in a soluble form and converting it to an insoluble form. So that was, of course, of interest to the Hanford site, potentially in the Department of Energy, for bioremediation purposes. Yep. Another bacterium that was the name escapes me at the moment. I think it's Deinococcus radiodurans. Yep. But that particular bacterium being resistant to radiation. So there was some early hope that they might be able to cross the two and have sort of a superbug to put into Superfund sites to help with remediation. So Department of Energy was interested, and we've just continued to build upon the technology, build upon the knowledge gain in different application space, and sort of bring it together to try and form, if possible, a more holistic understanding of the different ohms in any biological sample. It's something that I personally really enjoy about being at PNNL is, I get to see the perspective in applying these technologies and these approaches in the study of health and disease. Mm-hmm. And then we've got fundamental studies of microbial communities in the soil. And so being able to take what we learn in one system and apply it to another and vice versa is a really cool opportunity that I don't think exists in very many places.
That's awesome. No, that's really awesome. And you got me thinking, as you talk about some of, not just the normal sort of microbes in our environment, but when you start talking about some of these extremophiles like Deinococcus and all the other interesting species out there can do things for us. I think back because, I come out of the pharmaceutical industry. We forget that the majority of my former industry was created from interesting secondary metabolites from the natural world, and most of the really smart people will tell you that we've looked at a very small percentage of that. We're going to be getting, as we get into further into what you're doing at PNNL, we'll discuss sort of how small a slice of that we actually have looked at. But let's take a quick break at one of your really interesting papers that was entitled "Sharing and Community Curation of Mass Spec Data with Global Natural Products Social Molecular Networking." I found this fascinating because it brings in some of these themes like, hey, there's a lot out there. We don't know much about it, but we got to share some of these sort of these databases that we have just alone because they have so much potential tapped in them. Talk a little bit about this particular paper, if you will.
Sure. Yeah, that's right. So that was a paper that I happened to have the good fortune of being a co-author on. It was led by a Peter Dorstein at UC San Diego, and Peter had and still has a vision for democratizing science as much as possible, and also for making available all of these resources in terms of reference mass spectra that we all sort of have tucked away in our own laboratories and we utilize for our own research, but to a large extent we don't necessarily make available to the community. And so Peter's vision was to create a resource where those that were interested could contribute their own reference spectra to the GMPS resource. And then he and his team, including Ming Wang, who's now at UC Riverside, had the clever concept of performing an exercise of spectral comparison that they call molecular networking, and to build these networks of related spectra and using the relatedness of spectra to potentially help annotate unknown spectra that one laboratory might have, but then might be chemically related to a spectrum that was annotated by another laboratory. And so it's been a tremendous resource. It really cuts down on the exercise of what's referred to as dereplication. That's a common term in natural products, where folks that are exploring, say, what they think is a novel natural product from maybe a sponge in the sea, maybe someone else across the world had already discovered that, and they would like to know that it's already been discovered as a known compound before they go through the process of grind and find, as we describe it in that community. And so that's one of the purposes of the GMPS resources, to just enable individuals to annotate their data much more quickly, to speed the pipeline of annotation in these experiments, and to just utilize the resource that's out there in the world, but had yet before that time to be put together in a centralized resource.
Excellent. Well, I appreciate you walking us through that, because I really think it sets the stage for where we're going with this. And it was a couple weeks ago now that the PNNL article on your program, and a lot of what's going on, chemists are on the hunt for the other 99%. And again, this brought in, written by our friend Tom Rickey. This again brought back memories, because again, I spent a little time in the natural products domain myself, and I saw the HPLC and the mass spec and all that fancy equipment there in the lab. And when I asked that scientist to blast apart the grind and find, I would get the nice little spectrum, and oh wow, that's a nice spectrum. And then it was telling me that's a small percent of everything that's there. Take us on a little journey in terms of why the technology as it's currently set up, as we get to study our grind and find samples, only gets that little slice of the pie today, and a little bit of how this stimulated the thinking around the M over Q project.
So when I think of molecular analysis and using mass spectrometry, I mentally bin it into things that are essentially polymers that have repeating units of some number, like proteins or glycans. And then I think of the vast chemical space that really isn't governed by a template, if you will, in terms of the chemical structure. And it's the things that are polymers are much easier to identify using mass spectrometry, because it's a mathematically finite problem in terms of what the composition could be or should be. Take a protein, for example. You've got 20 potential amino acids. You already know that you're likely limited to just those things comprising the structure. Of course, they can be modified by post-translational modifications, including even AGEs. But if we know the structures of those post-translational modifications, that's something that we can account for when we search spectra. For other small molecules whose structures are really only governed by thermodynamics in terms of what constitutes a stable structure, the possibilities can almost be endless in terms of what you might be searching for in your data for a structure. And so in that vein, we as a community have had a lot of success in applying mass spectrometry, liquid chromatography coupled with tandem mass spectrometry to something like proteins or proteomics. And I feel that paradigm, as it's implemented right now for non-protein, non-peptide small molecules, it's a little bit of a round peg in a square hole. It's a really close fit, but it's not a perfect fit in terms of being able to allow us to identify every chemical substance that might be in a sample. And I've been in the field now for 20 years, and I've seen what we've been able to accomplish individually in our own laboratory using that paradigm, and I've seen what the community has been able to accomplish across the world. And we do make, I would say, steady linear progress in terms of new discoveries, discovering either new chemistry or in terms of the structures that are present, or new biology associated with those structures. But if you plotted that linear progress, it's got a pretty low slope compared to the fraction of chemical space that we've uncovered to date. And so I feel that we need to really disrupt the current analytical paradigm, not only instrumentally, but also computationally, if we are going to be able to identify chemically everything that's present in a sample. And then based on everything that's present in the sample, in terms of the chemistry, begin to uncover new biology associated with that, depending on the system that we're looking at. And so the article that you mentioned describes a new instrument that was developed at PNNL. Not 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 100% new, but new in terms of the two components that were coupled together. And so the first author, Adam Hollerbach, took the PNNL developed structures for lossless ion manipulations, which is an ultra high resolution ion mobility separation device, and he coupled it with a commercially available Orbitrap mass spectrometer, which is one of the best mass spectrometers on the market for performing high throughput omics measurements. And so together, he's now multiplied the overall resolution of each individual component. He's multiplied them together now to have an instrument that can perform a high resolution gas phase separation of ions coupled with the high resolution mass spectrometry measurement of the separated ions. And the other exciting component of the paper, which we of course didn't mention because there's another paper on the other topic, is we can computationally predict these high precision collision cross section values that are a parameter that's measured during the SLIM separation. We can computationally compute those. And there are also tools available for the computational prediction of the tandem mass spectra that the Orbitrap will measure. And so now, hopefully, in the years to come, we will be able to throw off this burden that we've all carried around with us for so many years of needing a pure substance in the bottle to definitively identify a compound in a sample. And coming back to the natural products analogy and grind and find, the way that folks identify new natural products is to isolate them from a sample and get enough quantity of it that you can submit it for an NMR structure determination. But sometimes it's difficult to get such quantities. And so if we could have a technological approach that would yield the confidence of an NMR based chemical identification, but on the scale of a mass spectrometry measurement, which would be, much less, potentially micrograms of sample, then that could really speed the process of new discovery.
I remember those days too. You don't have enough material. Come back when you do. I've been there as well. No, this is why this again. When I read that article, I was just I was jumping around because again, it took me back a few decades. But I'm it's just very exciting that you're working on advancing and developing these tools. Let's talk about a couple of other examples per. And again, for everybody that's listening and watching, you can find Dr. Metz's extensive publication record in Google Scholar, and just there's a wide variety of programs involved in. I think it's interesting to just start with let's see. This is Nature Biotech. No, this is Nature Microbiology. November 2022, standardized multiomics of Earth's microbiomes, revealing microbial metabolite diversity. Obviously, as you were mentioning before, the importance to understand a lot of these unknown microbes and their potential for enzyme production or bioremediation. We also contain a series of really interesting microbiomes, which has gotten kind of hot. And again, last couple of years said, hey, we need to understand a lot more about these trillions of things that are living within us. But then, as we know, they have proteins and lipids and everything else, and secreting all sorts of secondary metabolites. What are some of the interesting things, whether it's for the human health or, again, the energy front, that you look forward to in exploring with these new tools as it pertains to interesting microbiome samples?
Yeah. Well, certainly their impact on health and disease. I mean, the microbiome has been. It seems like if you conduct a proper study of the microbiome, you can find a role for it in almost any aspect of human life, whether it's circadian rhythm. We had previous publication with collaborators at Caltech on a role for the human microbiome in the development of autism spectrum disorder. So the gut microbiome is almost its own organ.
Yep.
It's not an organ in the context that it's a tissue or a proper organ like the pancreas or the heart, but the way that it functions, it's such a vital component of the gut, and limiting it to just the gut microbiome. Of course, we've got skin microbiome and nasal and oral microbiomes that all live with us in a either symbiotic or even pathogenic manner, depending on what you might encounter in your day-to-day life. And so that it's a avenue for the development of new therapeutics, whether it's probiotics, or even potentially pharmacological interventions where you might want to control a particular population of bacteria in the gut, or even enhance their numbers in the gut if they are performing a certain function, beneficial function, or detrimental function in the case of wanting to clear a particular organism out. And these interventions, I think, the more we learn about the microbiome, the more targeted we could be with the interventions, as opposed to just, say, taking an oral antibiotic that might target more than what we would want it to target, and clear out too much of the microbiome. And so I think we are in as much as has been done in research with microbiome and understanding its role in health and disease, I think we are in very early days of understanding the full impact and full potential. And there are very good things to come in the future as we learn more. It's a great opportunity.
Complete agreement on that front. Yeah, that's extremely exciting, not just from the novel uses of these sort of living health products, but at the same point, as you're pointing out, we can go beyond antibiotics, quorum sensing compounds, and all sorts of other interesting things that are ways to approach, especially in this era of antimicrobial resistance. So that's really exciting. Another one, Tom, that I found really interesting was a preprint. I think this is recent, actually. This is from just a couple months ago. Met Archives, and this is entitled "Systematic Review of Type 1 Diabetes Biomarkers Reveals Regulation in Circulating Proteins Related to Complement, Lipid Metabolism, Immune Response." And again, here, when we think about sort of the biomarker stuff that's going on, whether it's specific diseases or health indications, what have you, we kind of take a narrow approach nowadays. They think about, HbA1c. What does that tell us about the disease state? But as you sort of point out in this paper, look, disease ain't just one thing. There's a lot going on, and these fluctuations over time of these various metabolites and circulating protein and everything else, as in the case of type 1 diabetes, tells a picture of health and disease. Talk a little bit about what you're up to in this space, if you would.
Sure. Yeah. So we've got a number of projects in the area of diabetes, primarily type 1 diabetes at the laboratory. And again, it's, bringing the advanced omics analysis capability to bear to try and uncover new chemistry that then informs new biology. Our primary efforts are understanding as much as possible about the signaling events that occur in the human pancreas during the autoimmune stages of type 1 diabetes. Once the autoimmune process has set in and an individual has developed autoantibodies, it is essentially a guarantee that individual will develop type 1 diabetes at some point in their life. And so the opportunity space is really in the events leading up to the development of autoimmunity. If we can understand what these triggers are, and the triggers can include not only primary triggers such as, say, a viral infection that might initiate the cascade, the internal cascade, in terms of the immune response that ultimately lead to autoimmunity, or potentially these are environmental toxins that someone is encountering, man-made, for example. If we can understand, what exactly these triggers are, and likely there are many triggers, and likely there are differential susceptibilities among individuals based on genetic background to the different types of triggers, that makes it a really complex issue to unravel. And I think that's why we haven't developed an intervention. We haven't even discovered, what the triggers are ultimately that lead to autoimmunity. But we've got several projects funded under the Human Islet Research Network, which is a large consortium of other consortia funded by NIDDK that are performing a lot of fundamental studies and even translational studies to understand essentially the biological mechanisms that are occurring inside the beta cell, for example, that might lead to ultimate loss of insulin production and then eventual development of type 1 diabetes. But just a large set of projects within that consortium. We have maybe 2 or 3 of those projects right now, and just building up basic knowledge on how does an islet respond to cytokines, for example, that will initiate an inflammatory response? And are these processes and markers that we see, are they then replicated in a cohort of individuals with type 1 diabetes, which is our work under the TEDdy consortium, essentially biomarker discovery. And we do see correlations. We don't see exact matches between, for example, proteins that might be turned on in the beta cell due to stress. We don't see an exact match to what we see in the blood of individuals within TEDdy, but if we raise those proteins up a level into biological processes, we start to see some themes in biological processes like antigen presentation, which you might expect in a disorder that's initiated via an autoimmune response. And so it's, again, I think we've got a unique opportunity to be funded under both programs to perform fundamental studies and then to look to see if any of the fundamental changes and processes are then occurring in individuals who are at risk for developing type 1 diabetes within the TEDdy program.
Very, very cool. Tom, one other one I want to ask about, and this is September 2022 in the Journal of Cheminformatics, an initial investigation of accuracy required for the identification of small molecules in complex samples using quantum chemical calculation, NMR chemical shifts. Quantum obviously is kind of a hot topic nowadays. We've done a few shows, and I know quantum computing isn't here yet. There's a lot of early stuff, but and then we played around with the theme of quantum biology on a couple episodes. What role does quantum play in a lot of this? Obviously, when we break things down, they're atoms and fields and all sorts of other things, spins and how does sort of the whole theme of quantum filter into how you think about this new world of exploring the 99%, especially with the NovaQ project?
Thinking of quantum, it's a great question, because on the other side of the coin are artificial intelligence approaches that can, perform similar predictions of chemical properties, but much, much faster. And so that paper that you mentioned, there need to be more papers like that, because the community has here and there developed a variety of approaches for either artificial intelligence-based predictions of molecular properties or quantum-based predictions of molecular properties, but there hasn't been a lot of assessment of how accurate any of these predictions need to be in use cases. And when I think of implementing either of the approaches, I don't think of a graduate student project where there's a molecule being studied, whether it's a protein or small molecule, and the student has developed or is applying any of these predictive approaches to determine how accurately any of them can be in predicting a property, particularly for that one molecule. I think of applying these for everything that might be present in a sample, everything that we want to measure, and even going beyond that, let's apply them in computing properties for what we call the molecular universe, which might differ depending on who you talk to, but let's assume it's maybe, I don't know, 10 to the 60 molecules, which has been a speculation based on combinatorial approaches. So um, the way that we implement analytical measurements of these molecules experimentally, it all comes down to comparing experimental data to similar data that we've generated through the analysis of a pure substance in a bottle. And that is the single biggest limitation to unlocking, the knowledge that might lie within the chemical universe in terms of biological function. Most laboratories have about 8 to 10,000 standards in pure form at their disposal. And it's just due to budgetary limitations. Usually it's due to what chemical suppliers you may be aware of. Others have been able to assemble very, very vast reference libraries of pure substances, primarily through good relationships with biotech and pharmaceutical companies. But not everyone, has that luxury to, have those types of relationships. And so for me, I believe the future for molecular identification is heavily, heavily dependent upon computation, advanced computation, and computational prediction of the properties that we then measure experimentally. And so completely doing away with the development of reference libraries through analysis of pure substances, we need to replace that with development or construction of reference libraries through computational prediction, whether it be AI or quantum chemistry. I would love for it to be AI-based approaches because they're so fast, but they require a training set, and we're usually limited in the quantity of data that can be submitted for the training set. And at least with chemistry, with chemicals, as soon as we get outside of that finite training set, we start to see the models break down and the errors increase in terms of the predictions. With quantum, they can be very, very accurate, but it might take you 1,500 clock hours to compute a single property for a single molecule, which again, isn't necessarily feasible if we're going to unlock the secrets of the chemical universe. And so this question of evaluating accuracy in predictions, it needs to be done. It needs to be done on a grand scale because we need to know what we can get away with in terms of prediction errors. I also believe that trying to identify molecules based on a single property, like a tandem mass spectra, or an NMR spectra, or a retention time, that will never suffice. It needs to be multi-dimensional measurements, and it needs to be coupled with multi-dimensional predictions of the properties that are then measured in these multi-dimensional measurements. I feel that will begin to reduce the degrees of freedom, if you will, in the chemical space, and allow us to assign more uniqueness to the identifications that we make. We don't have an idea of really what it's going to take in either the dimensionality of the analysis or the accuracy in the predictions. That's something that we're exploring now under a project within the MORQ initiative. And I look forward to that because I would like to know, because it would be great to be able to set the bounds somewhere and sort of move on from the uncertainty in what we need in terms of predictions and dimensionality of analysis. And once we know, then we can just implement it. And it becomes a huge laser or mass spectrometry is already a massive hammer. We make it even more so, and then we just begin to pick the problems that we begin to apply the technology to. I think we can do it.
Awesome. That's really awesome. Again, it's going to be interesting to see you dump this 99% additional into those training sets, as you were mentioning with the AI.
Yeah,
it's really fascinating space. What just offhand, are there specific areas of chemistry? I mean, I remember, back in the day, and we talked about small molecules and proteins. I remember, 20 years ago, people wouldn't go near the, carbohydrate chemistry and all that stuff. It was what areas excite you? What areas scare you still? I mean, are there any interesting chemistries that you run into? You're like, wow, why does nature make this?
Yeah, well, I certainly think just the entire suite of chemistry, for example, that might be present in a soil sample
Got
and
it.
associated with a rhizosphere, the relationship with the plant, so complex. Of course, you have primary metabolism, which most organisms have a lot of overlap in primary metabolism, but then you
Sure.
have the secondary metabolism that's responsible for signaling of some sort, and maybe it's chemical warfare to ward off a competitor, maybe it's a stimulant to cause your next door neighbor to overproduce something that you then can utilize. But we've seen the data from some of the measurements conducted at the lab by colleagues using FTICR, and the chemistry is immensely complex in
Mhm.
terms of not necessarily the composition, elemental composition, but the number of molecular formula that you can calculate fairly accurately from an FTICR measurement of a soil sample is just tremendous. And it gives you an idea of the likely structural diversity that's present in just that one sample.
Yep.
So that I think is the system that scares me the most. And it's not just because of the diversity of the chemistry, it's because of the nature of the sample itself, the
Got
mineral
it.
content that may bind molecules irreversibly that you cannot extract out. And so
Got it.
there are likely blind spots in the analysis, things that are still there that are very
Mhm.
relevant, but just is not amenable to the typical sample prep
Right.
procedures that uh, get the constituents ready for an analysis. So
Gotcha.
I think in terms of the complexity of executing an analysis, as well as just the complexity of the chemistry within that sample type, that's the scariest, but also it's potentially one of our greatest opportunities, and especially in terms of carbon sequestration, because again, there are these minerals and other aspects of soil that bind carbon containing molecules irreversibly. And if we can fully elucidate the nature of the chemistry that binds irreversibly to some of these aspects of soil, then that becomes a sink that we might be able to utilize for carbon capture and thus
Yep.
address the growing climate issues that we have. Um, the bacteria in the soil are going to play a role. The plant, if we can understand the nature of the chemistry of all of the metabolism that's occurring in those relationships, maybe there's something that we can exploit in terms of funneling carbon into a form that will not be accessible for further metabolism and release back into the atmosphere, either via carbon dioxide or methane or um, other forms.
Really fascinating. I'm excited for you. I just really such an exciting portfolio. And wish you the best with all this time. It's just you see the potential from all angles: human health, energy, agriculture. So
Yeah.
just I'm rooting you on. Um,
Thanks. Yeah.
while we have you, anything else hot coming up for the rest of 2023? Any conferences or meetings or places that we can run into you,
Sure.
listen to you?
Yeah.
Please.
Yeah. So I'll put a plug in for the Metabolomics Association of North America, just because I'm the president
Nice.
at the moment. So that meeting will take place in October on the campus of University of Missouri, and there should be some of the latest advancements being presented in metabolomics and molecular analysis there. I think the dates are October 23rd through the 27th. So check it out. The conference web URL is mana2023.net.
Okay, we'll put a link to that. Perfect. Awesome.
And
All
then for
right.
our own research, there will be some interesting stuff coming out. Maybe if I had to speculate, and depending on how peer review goes, potentially around October, November timeframe, we've got some really cool observations on fentanyl chemistry coming
Mm,
out of the SLIM Orbitrap instrument, and there's some there's some cool nuggets in there that I don't want to say any more on to not steal the thunder from the author of that paper.
Got it. We'll be watching for that one, because that is clearly a very timely topic as well, one that we wish we have to deal with, but
wish we didn't have. Yeah,
yeah,
that's for sure.
But no, that's fascinating. Really great stuff, Tom. Again, I really appreciate you telling the story. For everybody that's going to be listening to this particular episode of the show across the various podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Thomas Metz, senior scientist, laboratory fellow, principal investigator, integrative Omics Group, Biologic Sciences Division at Pacific Northwest National Laboratory. Tom, again, thank you for taking the time out of your schedule to come educate us on all these themes. Obviously, thank you for everything you're doing there at PNNL. And as we like to say here on our show, thanks for helping you create a better tomorrow via the type of work you do. Really great stuff.
Thanks, sir. Appreciate it.
Great having you.